The Bogata Research Institute has been establishing a new grapevine improvement team. We're based here at Lincoln University, where we're tenants, and we have a new laboratory that does a lot of genetics, a lot of molecular work, understanding the differences between different varieties of grapevine and different clones of our premium variety, Sauvignon Blanc.
In our research vineyard, we have 6,000 unique SB clones. Specifically, we are focusing on primary key industry traits such as disease resistant, high yield, drought resilient, including water use efficiency. Our goal is to identify the most effective way to screen this larger population of clones.
We developed this pool of new Sauvignon Blanc clones with increased genetic diversity, and we're doing it by harnessing the plant's natural ability to adapt. We treat them with stress, and responding to that, the plants will generate random genetic changes. So we do not introduce any new or foreign genetic material, so they are completely non-GMO. Traditionally, we select vines by looking at the traits in the vineyard, which sometimes can take years to observe. But with the advance in sequencing technology and genome analysis, we can now look at the genetic information of the vine that might predict some of these traits. We have our own sequencing in the lab, which is the Promethean P24 long-read sequencing that we use to sequence these plants. We can find a pool of genes that have been correlated with certain traits, and we can look at specifically at the targeted regions.
One of the major challenges to understand the resistance of powdery mildew of this many clones of the 1,000 plants. We have a microscopy image robot in the lab. It has a computer vision, heavily relies on machine learning and AI algorithms. So we feed leaf discs inoculated with the powdery mildew into the machine, and it scans through all the discs and then gives us a score which discs have the percentage of infection. We can screen at least about 1,500 discs in a day, whereas if we do it with the microscope, it will take at least 2 months to do that sort of work.
At the moment, we are using 80 subset of SB2 Sauvignon Blanc in the glasshouse trial to monitor the drought resilient. And the aim is to upscale those traits that will be consistent across the different clones in our research vineyard. We are using the 4th generation photosynthesis meter to monitor the gas exchange in leaf, and also to monitor how this plant can photosynthesize under drought stress. This is just the start of the trial, still a long way to go to identify this physiological and biochemical trait.